Hey, it's Amy with Behind the Tweet. Today I want to talk about the human virome. You've seen me tweet a couple articles or papers on the virome. I think the virome is one of the most exciting areas of research happening at the moment. Let me give you some more context. Most of us know that there's a bacterial microbiome in the body, especially in the gut. I think most of us are familiar with the fact that trillions of bacteria persist in the gut, contribute to health and disease processes. Now, I've also encouraged you in my papers, and many other research teams are showing that the microbiome extends beyond the gut. In fact, it appears to persist in most tissue and blood. This is the bacterial microbiome. So most of our human tissue and blood has bacteria. There's a lung microbiome, a bladder microbiome, you name it microbiome at this point, right? So what the virome refers to is the fact that we've been taking some of the same computer-based technologies that we've been using to better study bacteria in the bacterial microbiome, and we've been turning it on the study of viruses in the human body, new tools. And while using these new computer-based tools, we're beginning to discover that there are vastly more viruses in the body than we previously thought. And those ecosystems of viruses that also persist in almost all human tissue and blood are referred to as the human virome. Now, most of the virome includes all viruses in the human body. So Epstein-Barr virus or the herpes viruses, or any virus that you probably know well and have heard about being linked to a chronic disease or something you suffered from. Those viruses can persist in the body often. And if they do, they're part of the virome. The virome consists of RNA viruses, DNA viruses, any virus in the human body. But what we're realizing is many of the viruses in these ecosystems are what are called bacteriophages. Bacteriophages are viruses that infect bacteria. So they directly infect bacteria, they insert their genetic material into the bacterium, and by doing so, they significantly modulate the activity of that bacteria, what it can and cannot do, and whether it can even survive and persist in the body. So these bacteriophages, or phages for short, phages control a large amount of the bacteria, the activity of the bacteria in the human body. And that ability of these viruses to control bacterial activity is one of the things that makes these human viruses, or phages, most relevant to our understanding of chronic inflammatory disease. Because for a while, we've been tying the bacterial microbiome to chronic inflammatory disease, or we've been tying imbalance of the bacterial microbiome to it. And now we can begin to include the virome into that picture. Before I go on further, let's just talk about the virome. How many viruses are we talking about? Well, we're talking about many of them. So today, I tweeted an article written by David Pride and one of his colleagues. It was on a website called The Conversation. And David Pride is the Associate Director of Microbiology at the University of California, San Diego. And he references a study in that article which estimates that there are over 380 trillion viruses inhabiting us. This community collectively is known as the human virome. So, okay, over 380 trillion viruses in this virome that we're beginning to understand plays a major role in the human body's microbial ecosystems, or organism ecosystems. And David Pride went on to say, quote, "Viruses may inhabit all surfaces, both inside and outside of the body. Everywhere researchers have looked in the human body, viruses have been found. Viruses in the blood? Check. Viruses on the skin? Check. Viruses in the lungs? Check. Viruses in the urine? Check. And so on. To put it simply, when it comes to where viruses live in the human body, figuring out where they don't live is a far better question than asking where they do." I think that is a great quote from David Pride, totally relevant. These trillions of viruses appear to persist in all of the human body, important players in our ecosystems. Okay, so I'll reference another paper along those same lines. It's a paper called Bacteriophage Transcytosis Provides a Mechanism to Cross Epithelial Cell Layers. Again, the paper stresses that, quote, "Bacterial viruses are among the most numerous biological entities within the human body. These viruses are found within regions of the body that have conventionally been considered sterile, including the blood, lymph, and organs. So this study used several laboratory studies to show that there appears to be rapid and directional crossing of these bacteriophages between different cell layers in the human body. Essentially, these bacteriophages are very good at crossing cell layers that separate different organs or tissues in the human body, which means it seems easy for them to traffic throughout the entire human body. So he goes on to estimate that 31 billion bacteriophage particles cross across the epithelial cell layers of our human bodies every day, of the gut and into the average human body each day. And he was looking mostly at bacteriophages that can enter the gut because, of course, like anything, nothing in our world is sterile. There are so many organisms, all kinds of organisms, and everything that we consume, touch, every part of our world. So there are bacteriophages in our food, there are bacteriophages in our water, just like there are bacteria, just like there are other organisms. So every day we are picking up bacteriophages from our environment, in addition to the ones that already persist in our communities. And all of these bacteriophages, whether we pick them up that day or this or not, they're all trafficking throughout our system. So just take a second right now and picture 31 billion bacteriophages, or a sea of bacteriophages, as they have been referred to, circulating throughout your tissues and blood. Well, it's very clear, I think, that the body is not sterile. The opposite is true. So sometimes when I tell that to people, they're like, that is a really cool fact. Tonight at dinner, I can't wait to tell my friends that there are 31 billion bacteriophages. And some of my friends are like that. And I'm like, that's actually cool. I'm really glad you're excited about the topic. But obviously, we need to go beyond numbers here. If there are trillions or billions of viruses, mostly bacteriophages, but all of these viruses in the human body, then clearly they have a significant impact on human biology. And clearly, there's no way they're not doing anything. They must be doing a lot of things. What are those things? How are they acting? And how does that contribute to human health and disease processes? Well, here's a major way. So far, we've been studying the bacteria in any microbiome community. So let's say we look at the gut microbiome. There have been, hundreds and hundreds of analyses now that look at the bacterial microbiome in patients with an array of different chronic inflammatory conditions. At this point, almost every human chronic inflammatory condition is tied to gut microbiome dysbiosis at least, if not dysbiosis or imbalance of the bacterial microbiome in different body sites. But since the bacteria in these microbiomes are modulated by the activity of the phages and other viruses in the virome, and of course, these communities are persisting together. The virome is not separated from the bacteria. They're all continually interacting, and they're interacting with fungi and archaea too. This is one big ecosystem. These bacterial microbiome studies have not been taking into consideration the fact that viruses in the same area are very likely modulating the activity of the bacteria identified in the study. So in some of these bacterial microbiome studies, or in the majority of them, we're missing a large part of the picture because we haven't been studying what the viruses in the same ecosystems are also doing. Now, if I could try to simplify that with an analogy, maybe I would compare the bacterial microbiome or bacteria in a microbiome community to students in a school. So you could look at a school, let's say we're observers and we'd never seen a school before, and we started observing the students. Consider those students to be like the bacteria in a microbiome community. We're looking at them, we're saying, wow, look, they all tend to sit down at the same time in their desks. They look up at the blackboard. They're quiet for long periods of time, but then they sometimes get up and they change rooms and they sit back down again. Very interesting behavior. And we'd come up with a lot of theories as to why the bacteria were acting that way. But the viruses, or the virome, that we're now beginning to study could be compared to the teachers in that same school. It turns out that the reason the students are sitting down, looking at the blackboard, doing a lot of the things they do, getting up between classes, happens to be because the teachers are making them do it. They're saying, you need to sit down, you need to be quiet, You can get up when the bell rings. You can move. They are modulating the activity of the students in the classroom. So in order to understand how a school is functioning, we need to understand the students and the teachers, their relationship with each other, and then we can better understand the dynamics of that community. The same goes for our ecosystems of microbes and viruses in the human body. If we only study the bacteria, we will not understand how viruses are modulating their activity. So now that we're better characterizing the virome, we have this amazing opportunity to better see a full picture when it comes to microbiome studies. We can factor the viruses more into the picture, and by doing so, we can gain a lot more information. So let me give you an example. There's a team at the Joint Genome Institute. A member of that team is David Paz-Espino, who I recently interviewed on my blog. You should check it out. And they published a study called Murine, or mouse colitis, reveals a disease-associated bacteriophage community. So what they did is they took mice who had developed colitis, which is similar to irritable bowel syndrome, and they sequenced the bacterial communities in the guts of those mice, but they also sequenced the viral communities. So they had info on both bacteria and viruses. When they did that, they found that there were many, many phages in the guts of these mice, and that the phages did seem to be affecting the dynamics of the community. So first of all, if you look at the study, it's just really cool because by looking at the phages, you can see what phages are actually there. So there are phages in the gut of these mice. They found phages that infect lactobacillus. They found phages that infect salmonella. They found phages that infect E. coli. So that in and of itself is very interesting because those phages are there primarily because those are bacteria that they often infect, and we can better understand what bacteria may be there because the phages give a clue. We're getting better community dynamics is my basic summary there. In this study, going back to the details though, they found a decrease in phage community diversity in the mice with colitis, but they found an expansion in subsets of certain phages that were also in the mice with colitis. They found that some phages expanded, and some of these phages that expanded in number were phages that infect bacteria that are pathobionts or pathogens tied to colitis or irritable bowel syndrome. Pathogens that we know are bigger players in these conditions. What that seems to indicate is that the phages that might be infecting these bacteria capable of driving symptoms and inflammation may be playing a role in why those bacteria drive symptoms and inflammation. And overall, when they looked at the general dynamics of the phage communities, they found that in the mice with colitis, the community shifted from an ordered state to a very disordered and imbalanced state. So there was just a change in the entire way that the phage community seemed to persist in these mice with colitis. So obviously, we can do a lot more follow-up studies to better understand what's going on, but this is a great study because all of a sudden we have so much more information to run with on what is happening in the guts of mice with colitis. And by the way, the phage populations that they found in the mice that changed a bit in the colitis mice, while they did find that humans harbor many of those same phages. So this study probably has pretty big implications for human bowel disease in addition to just what they found with the mice. So that's really important. We can better understand these community dynamics by understanding what the viruses are doing. So there was a similar study done by researchers in China that I'll link to at the bottom of this talk. It's called A Human Gut Phage Catalog Correlates the Gut Phagiome with Type 2 Diabetes. And sometimes the phagiome is used to refer to just the phages in the virome as opposed to all the viruses in the virome. So again, in simple terms, they identified in humans a type 2 diabetes specific gut phagiome, which was basically, again, the patients with type 2 diabetes had different phage composition or the ecosystem community of the phages was different in the patients with diabetes enough that it seemed like it was a distinct community of phages in those patients. They'd have to do more follow-up studies, but that's a very interesting early finding. So moving on from that, there's a researcher named George Tetz, who is the head of the Human Microbiology Institute in New York. And I've read several of his papers, and I listened to him interviewed today by David Perlmutter, who is a neurologist, I hope, that actually communicates a lot of research to the general public, which I really respect. And so that interview, and I had already read, I encourage you to listen to it, I'll link to it, goes into some of George Tetz's work that I find to be interesting. He is actually looking at bacteriophages as new human viral pathogens. In fact, he just published a paper called exactly that, Bacteriophages as new human viral pathogens. And what he did was propose that bacteriophages may not only infect bacteria in the body, but that they appear to also directly and indirectly interact with human cells and human proteins. He's not proposing this off nothing. If you read the paper, there's actually a long history of researchers who have studied bacteriophage behavior, going back to the turn of the century. We've known, actually, that bacteriophages persist in the human body for a long time. We just haven't been studying them, and we certainly haven't been studying them with the latest technologies that we've just begun to start using more. So he references a number of studies which raise an interesting possibility, which is that bacteriophages may also modulate the activity of our human cells. If they do that, they probably also, almost by definition, can influence the human immune response. He goes into that too. It appears that phages can also influence how the immune system acts. So now that brings in another component to understanding the virome and the phagiome. Because sometimes if we see the immune system in the body acting interestingly, we may now have to factor in the consideration that phages may also be impacting the immune response. Again, this is really early stuff, but these are excellent questions to be asking. So he goes, Tetz, into the paper. He goes a little bit into how phages may be affecting neurodegenerative diseases, the pathogenesis of such diseases, and also autoimmune conditions. And to give you a more specific example, he recently published a paper called Parkinson's disease and bacteriophages as its overlooked contributors. So this study looked at the association of bacteriophage communities in the guts of patients with Parkinson's disease. And he identified in several of the subjects, and he could also include some more subjects. This needs to be done in more patients to be verified, of course. But he did, in some of the Parkinson's subjects, identify a shift in the phage to bacteria ratio of lactic acid bacteria known to produce dopamine and regulate intestinal permeability. So that's one interesting thing. He's been finding, again, early findings, that phages may have an impact on intestinal permeability. So when we're talking about leaky gut or syndromes like that, it looks like we might have to include phages in the picture of why the barriers in our gut might become more permeable. That's something that George Tetz is looking into, which I think is very interesting. But going back to this Parkinson's study, he basically found that there are certain bacteria in the gut that produce dopamine, lactic acid bacteria. Now, the activity or concentrations of those bacteria can be modulated by certain phages. And though that may cause a shift in how many bacteria are able to produce dopamine. Now, that would affect patients with Parkinson's disease, since dopamine is a neurotransmitter that plays a role in Parkinson's disease pathogenesis. So again, studying the virome adds a layer to all these chronic conditions, understanding how microorganisms are involved, because we can look at what the bacteria do, we can look at what the phages do, and we could look at the end product of those interactions and how they may affect the disease process. In this case, there may have been modulation of dopamine, but that modulation was a mix of what the bacteria did and how the phages were regulating the bacterial behavior. So these are really cool studies. And I want to emphasize then that I hope that more researchers continue to study the human virome. Right now, if you study single viruses, you're not studying the virome. This is something I want to make clear. There's nothing wrong with the study of single viruses like Epstein-Barr virus or HHV6, but it's important to better understand how these viruses persist as members of communities. Because think about it, I am not a lone person. Every day, my behavior becomes the sum of what I do, how I interact with other people and the surroundings around me. That becomes the sum of my activity in any one day. It's the same for these viruses. Epstein-Barr virus, for example, does not likely persist all by itself all the time. It is a member of the virome. And so it may very well interact with other viruses and of course bacteria in the communities in which it often persists. It becomes a member of the virome when it persists in the human body. So if we better characterize this more extensive viral population in the body, we may better understand how some of these more well-characterized, well-studied viruses that we're also already familiar with, we may better understand their activity and their ability to drive disease processes. So, and of course, many of these viruses in the virome have simply not been characterized, named. They may in and of themselves be pathogens too. So really, we're gaining a much bigger picture of what organisms in the body may be able to interact together or even alone to drive disease processes. So I want to make clear, a study that just uses PCR, for example, as a tool is not studying the virome. PCR as a technique can only search for known viruses that we already know exist in the human body. So in order to study the virome, we have to use some of the new tools and pipelines and systems that specific research teams are using and creating right now. So my hope is that more research teams begin to use those technologies to look at these wider viral communities in the human body. My hope for that is because I have always thought that with so many conditions still of unknown cause, so many human conditions, so many rare diseases that are still deemed mysterious and unknown in cause, could be connected to the microbiome and the virome. Think about it. So many conditions that we don't understand, but that are tied to inflammation. The immune system seems to be affected by something. And then an explosion of research on bacteria in the human body, fungi, and now on viruses. When all that information is taken together, it does seem more complicated. Yes, it adds to the complexity, but it also adds to the knowledge we may have to better understand what is happening in so many of these medical conditions that we do not yet understand. That is why I'm so excited about this research, one of the reasons. All right, that's it for now. Take care. Talk next time.